Generally, immunity refers to the resistance to infectious disease. The immunity is very important as the immune system is the body's first line of defense. It also protects one from viruses, bacteria, and other pathogens. Without a healthy immune system, one could easily fall. A good nutrition, regular exercise, and managing stress are fundamental to improve immunity. So, what is immune system? Immune system is the collection of cells and tissue that protect the body from infection. It helps to protect the body from harmful disease and keep us healthy by distinguishing our normal cells and tissue with other pathogens such as bacteria, viruses, and fungi. The immune system begins to develop in the embryo, and by the time the baby is born, it is respond to any antigens whether it is harmless or harmful. Everyone's immune system is different and it reacts differently to every antigens. Lymphocytes are a type of white blood cells that are playing vital role in immune system. They are found in the blood, lymphoid tissues, and most organs of the body that express receptors from specific antigens and mediate immune responses. Lymphocyte is divided by two, which is T cells and B cells. T cells or T lymphocyte is a type of lymphocyte that plays a central role in cell-mediated immunity that protects us from infection. It can wipe out infected or cancerous cells. It also directs the immune response by helping B lymphocyte to eliminate invading pathogens. B cells or B lymphocyte is a key player of the adaptive immune response that create antibodies for humoral immunity in mammals. The created antibodies will bind to pathogens such as toxins to neutralize them. B cells also can recruit other cells to help in destroying an infected cells. For immune system, it can be classified by two, which are innate immune system and the adaptive immune system. Both will work together to defend the infectious. The innate immune system is the first line of defense. It involves the barriers that keep harmful materials from entering the body. The properties of innate immunity are non-specific, natural, and native immunity. The innate immunity consists of cells and proteins that are always present and ready to mobilize and fight microbes at the site of infection. The examples of innate immune system includes cough, mucus, stomach acid, and so on. The role of innate immunity is stimulating adaptive immune response. Adaptive immunity is based on a specific response to a specific pathogen once a pathogen has breached the innate immunity defense. Comparing to innate immunity, adaptive immunity is lower response but it have a memory component. It can provide long-lasting defense and protection against recurrent infections. When adaptive immune system is exposed to a new threat, the specific of the antigen are memorized, so it prevents one from getting the disease again.
Hi, my name is Sarah and I will explain about the B lymphocyte structure. B lymphocyte structure have a unique structure that allows them to recognize and respond to the specific antigen. They are characterized by the presence of the surface receptor called B cell receptor, which we will call BCR. They are composed of two heavy chains and two light chains. For example, when a BCR binds to an antigen, it triggers a series of events that lead to the activation of the B cell and the production of the antibodies. This is the cell membrane of the B cell, which is a thin flexible layer that separates the interior of the cell from the outside environment. It is composed of a lipid bilayer which consists of two layers of phospholipid molecules, the hydrophobic and the hydrophilic. The cell membrane also contains various proteins including B cell receptors that are involved in antigen recognition and signaling molecules that help to transmit signal from the BCR to the interior of the cell. The cell membrane is selectively permeable, meaning that it will allow certain substances to pass through while blocking others. This allows the B cell to control the movement of molecules and ions into and out of the cell, which is important for maintaining the proper balance of nutrients and other molecules inside the cell. Net is the nucleus of the B cell, which is a round or oval-shaped organelle that contains the cell genetic material in the form of DNA. It is surrounded by a double membrane called the nuclear envelope, which contain nuclear pore that allow molecules to move in and out of the nucleus. Inside the nucleus, the DNA is organized into structure called chromosome, which contain the genes that code for all of the cell proteins. The nucleus also contains a nucleolus, which is then regions where ribosomes are produced. The ribosome then move out of the nucleus and into the cytoplasm, where they synthesize protein based on the structure encoded in the DNA. The nucleus play a critical role in controlling the activities of the cell, including gene expression and DNA replication. The cytoplasm of a B cell is the fluid-filled region of the cell that surrounds the nucleus and is enclosed by the cell membrane. It contains various organelles including mitochondria, ribosome, and endoplasmic reticulum, the Golgi apparatus, and lysosome. The cytoplasm is where many important cellular processes take place, such as protein synthesis, energy production, and intracellular transport. B cells are specialized white blood cells that produce antibodies, and the cytoplasm of the B cell is also where antibody production takes place. When a B cell encounters an antigen, it undergoes a process called activation, which triggers the production of the antibodies that are specific to that antigen. These antibodies are synthesized in the cytoplasm and then secreted out of the cell to help neutralize the antigen. B cell receptor (BCR) are specialized proteins that are located on the surface of the B cell. They are composed of two heavy chains and two light chains that are arranged in a Y-shaped configuration. The tips of the Y-shaped molecule are called the antigen binding site, which are the parts of the BCR that recognize and bind to the specific antigen. When a B cell encounters an antigen that matches its BCR, the BCR binds to the antigen and triggers a signaling cascade that leads to B cell activation and antibody production. BCR are essential for the immune system to recognize and respond to a wide range of pathogens, including bacteria, viruses, and fungi. Next is disulfide bridge, that are covalent bonds that form between two cysteine amino acids in the protein. Disulfide bridges are important for stabilizing the structure of the protein, including the B cell receptor. The heavy and light chains of the BCR are held together by the disulfide bridge, which help to maintain the overall shape of the receptor. Disulfide bridges are formed when the thiol groups of two cysteine resides come into close proximity and a chemical reaction occurs that results in the formation of a covalent bonds. Disulfide bridges are particularly important for proteins that are secreted outside of the cell, as they help to protect the protein from the degradation by the enzyme in the extracellular environment. The light chain and heavy chain of a B cell receptor is two of the chains that make up the receptor. The light chain is also responsible for transmitting signal into the B cell when the antigen binding site of the BCR bind to an antigen. The light chain is critical for the immune system to recognize and respond to a wide range of pathogens. The heavy chain is responsible for the antigen binding site of the BCR, which is located at the tips of the Y shapes. The heavy chain is also responsible for transmitting signal into the B cell when the antigen binding site of the BCR bind to an antigen. The heavy chain is critical for the immune system to recognize and respond to a wide range of pathogens. The signal transduction region of a B cell receptor is the part of the receptor that is responsible for transmitting signal into the B cell when the antigen binding site of the BCR bind to an antigen. The signal transduction region is located in the cytoplasmic tips of the BCR heavy and light chains, and it is composed of a series of protein domains that interact with intracellular signaling molecule. When the antigen binding site of the BCR bind to an antigen, it will trigger a conformational change in the receptor that lead to the activation of the signal transduction regions. This, in turn, will lead to the activation of a series of signaling pathways that culminate in the activation of the B cell.
Hi, my name is Alin Shraya and I will continue about the B cell development. Our cells will undergo lymphiosis which is development of diverse set of lymphocytes with unique antigen receptor. To become a B cell, it has to develop into an immature B cells in a bone marrow, and then complete its maturation into an antibody secreting B cell called the plasma cells in the lymph nodes and spleen. B cell receptor has two chains, a heavy chain and a light chain. To have a fully functioning B cell receptor, a B cell has to get through a series of successful genes arrangement. B cells go through 6 stages of its development. First, they started as common lymphoid progenitor cells. Common lymphoid progenitor cell expresses recombination of activating gene, which is RAG1 and RAG2, becoming early pro-B cell. Then it's late pro-B cells, heavy chain D and J gene segments spliced together via allelic exclusion as one S chromosome to complete splicing process. After that is large pre-B cell. Late pro-B cell attaches DJ gene segment to V gene segment via V-TJ recombinase binding site, which is heavy chain recombined with MU gene, which becomes large pre-B cell. MU gene codes for IgM constant region protein. Then is small pre-B cells. Functions as of heavy chain tested by binding to surrogate light chain V pre-B lambda 5. If it is successful, cells will be proliferate into small pre-B cell. Then, finally, immature B cell. Light chains are rearranged. Functionality of light chain retested by autoimmune regulator, which will identify self-reactive cells by expressing bodily antigen in lymphoid organs. As the cell develops, it makes permanent changes in its DNA so that by the time it's an immature B cell, it has DNA cell that uniquely codes for a B cell receptor that combines a foreign integer as the cell develops.
Hi, I'm Siti Zahra, and as for my part, I will be discussing the general functions of immunoglobulins, they're also known as antibodies. Okay, now let's explore the various functions of immunoglobulins, including antigen binding and effector functions. Firstly, antigen binding. The primary functions of immunoglobulins is to recognize and bind to specific antigens, which are foreign substances that can trigger an immune response. If you look at the picture of antibody right here, you can see that antibodies possess antigen binding sites that match the unique shapes of antigens. This recognition and binding process allow the antibodies to target and neutralize pathogens, preventing their harmful effects. Secondly, effector functions. There are 6 functions I listed up here, and one of it is agglutination of microbes. So immunoglobulins have the ability to bind to multiple antigens on the surface of microbes, causing them to clump together or aggregate. This aggregation makes it easier for other components of the immune system, such as phagocytes, to recognize and eliminate the microbes. Moving on to neutralization of microbes and toxins, immunoglobulins can directly bind to microbes or toxins, preventing them from interacting with host cell and causing harm. By neutralizing these harmful substances, immunoglobulins help to limit the spread and damage caused by the pathogens. And besides that, immunoglobulins can also access opsonin, which means they coat the surface of microbes, marking them for recognition and ingestion by phagocytic cells, such as macrophages and neutrophils. This process known as opsonization. It enhances the efficiency of phagocytosis and facilitates the elimination of microbes. Next, antibody-dependent cellular cytotoxicity. Immunoglobulins, particularly IgG, can bind to specific antigens on the surface of target cells, such as infected cells or tumor cells. This binding recruits immune cells such as NK cells. The NK cells then release toxic substances that induce the cell death in the target cells, leading to their elimination. Other than that, IgE antibodies can also trigger degranulation of mast cell and basophils. This process releases stored granules containing molecules like histamine, cytokines, and enzymes, which contributes to allergic reactions and immune responses against pathogen. And last but not least, complement activation. Immunoglobulins, particularly IgM and IgG, can initiate the activation of the complement system, which is a group of proteins that work together to eliminate pathogens. The binding of immunoglobulins to antigens triggers a cascade of events that ultimately results in the formation of membrane attack complexes, MAC, on the surface of pathogens, leading to their lysis or destruction. And all of these functions collectively contribute to the immune system ability to recognize, to target, and eliminate pathogen and harmful substances.
Hi, my name is Nur Akila Megumi and I will continue presenting on the topic B cell activation. Before we started, let's recall the sequence of events. Stem cells produce lymphoid progenitors and pro-B cells in the marrow just under the bone. When B lymphocytes mature and get activated, they will become plasma cells which secrete antibodies. B cell activation is also known as class switching because it involves the process of changing B lymphocytes from naive to mature. As we can see in the diagram on the slide, before activation, B lymphocyte can only secrete IgM and IgD, while after activation, B lymphocyte can secrete all types of immunoglobulin, includes IgA, IgG, and IgE. B cell activation is initiated when its receptor recognizes an antigen and binds to it. However, in most cases, B cell activation is dependent on stimulation by an activated helper T cell. Activation is carried out through a cell-to-cell interaction that occurs between a protein called the CD40 ligand, which appears on the surface of the activated helper T cells, and the CD40 protein on the B cell surface. The helper T cell also secretes cytokines, which can interact with the B cell and provide additional stimulation. Antigens that induce a response in this manner, which is the typical method of B cell activation, are called T-dependent antigens. Interaction with antigens causes B cells to multiply into clones of immunoglobulin secreting cells. Then, the B cells are stimulated by various cytokines to develop into the antibody-producing cells called plasma cells. Each plasma cell can secrete several thousand molecules of immunoglobulin every minute and continue to do so for several days. A large amount of that particular antibody is released into the circulation. The initial burst of antibody production gradually decreases as the stimulus is removed. For example, by recovery from infection, but some antibody continues to be present for several months afterward. The process just described takes place among the circulating B lymphocytes. The B cells that are called memory cells encounter the activating antigen again. These B cells rapidly respond by dividing to form both activated cells that manufacture and release their specific antibody and another group of memory cells. So, for example, if the antigen is microbial and an individual is reinfected by the microbe, the memory cells trigger a rapid rise in the level of protective antibodies and thus prevent the associated illness from taking hold.
In conclusion, immune system is playing a vital role in a body and they are needed by everyone. It protects one body from harmful substances, germs and cell changes that could turn to ill. It is important to strengthen the immune system. As long as the immune system is functioning properly, one do not notice it is working. However, when it becomes weak and unable to fight certain antigens, one can fall sick. That is why one should make an effort to strengthen their immune system. The benefits of having healthy immune system are one can once heal quickly, feeling strong and healthy, having a few infections only, and many more. In order to keep a healthy immune system, one should eat a nutritious diet, engaging in regular exercise, getting adequate sleep, get hydrated, and others.